The next team that we have that is going to be presenting is called RePair Genomics. RePair Genomics uses AI to design highly targeted gene control elements that let therapies turn on in the right cells and stay off everywhere else. And in order to present, we have Luca from the team. Luca, over to you.
My name is Luca, and today I'm excited to tell you about our technology at RePair Genomics. We hear about the potential for gene therapies, but rarely ever see any on the market. There are over 1,000 gene therapies currently in clinical trials, but only about 10% of these actually reach the market. Why is that? You see, it takes scientists months to design multiple drug candidates and then years to test them in animal models. Yet these drugs often fail in human patients, either due to low efficacy or high toxicity. In the end, it's not only years of R&D work, but also up to billions of dollars wasted per project. Luckily, RePair Genomics can change that. At our heart is our proprietary machine learning software, which was trained on next-generation sequencing datasets to design synthetic DNA sequences called promoters. These promoters act like a small on or off switch, telling the drug to activate only where it's needed. Using our promoters can not only increase the drug efficacy, but also reduce toxicity in both animal models and human patients. The gene therapy R&D cost per drug is over $1.9 billion US. And our promoter designs are not only limited to the gene therapy development space, but also towards developing transgenic animal models, biological tools, and use in genetic analysis markets. This brings our total TAM, SAM, SOM values up to over 23 billion, 17 billion, and 70 million US dollars respectively. From our recent data, we were able to show that RGNeuroV1, our synthetic neuron promoter, targeted mouse neurons specifically, and was 3 times stronger than the current commercially available neuron-specific promoter, while being less than half the size. Apart from the data generated in-house, we've also received confirmation from one of our collaborators, where our brain-targeting promoters, CNSA and CNSB, were more selective to the brain as seen through this in vivo live imaging than the control, which also showed significant peripheral targeting as well. Our competitive advantage lies in our speed, validation data, and purchase options. We'll have a catalog of generic promoter designs along with validation data available on our website for direct licensing. And additionally, we'll have the option to co-develop and custom build a promoter for our client, offering more targeting nuance than the generics in our catalog. Our competitors use a manual high-throughput screening method that would take up to a year to yield candidates ready to ship. While with our approach, we'll be able to generate candidates in 2 weeks to 3 months, depending on the validation data requested. More importantly, we also have a much more flexible price range to serve biotechs and pharmas of different sizes, unlike our competitors who are fixed to a customized order. Our plan is to first launch our promoter catalog. This allows clients to license and use our products as a first pass before diving into a custom order if they need a higher degree of targeting. Our strategy behind this generics catalog is twofold. First, it offers our clients and potential clients immediate access to our primary data. And secondly, it allows purchasing via license options for both large and small clients alike. Additionally, we'll also offer enhancer sequences as add-ons to our promoters. These act like booster packs in gene therapies to increase the strength of the drug. And so, using our designs can save our potential clients months of time and hundreds of thousands of dollars per project in the short term, while in the long term, we're saving them at least $200 million and years of R&D work based on the articles we found about failed gene therapy projects. Our primary clients are R&D teams in biotech and pharmaceutical companies working on gene therapy development. And so far, we've established collaborations with academic labs and biotech companies across North America. In addition, we are actively in discussion with several other potential partners, which reflects both the demand for our solution and the trust we are beginning to establish in the field. We've conducted a market survey analysis through BioTalent, where industry and academic respondents were asked about their greatest challenges in the gene therapy development process and what their needs are. On average, it takes organizations months to complete the first set of drug candidate screening before initiating any animal studies. So our service can significantly reduce the time with a streamlined process. We also noticed that most organizations are hesitant to switch to another promoter design process because of the time and effort required to revalidate these promoters. We can address this by working with academic labs to have them validate our promoters and publish the data together, increasing the credibility of our products. So far, we've developed our machine learning models and conducted internal validation experiments. We've secured pilot users, lab space at the Spin-Up Incubator in Mississauga, and raised over $500,000 in non-dilutive funding. And as of Q1 of this year, we filed our provisional patent for our catalog on about 40 different promoter sequences, targeting a breadth of cell types and organ systems. This year, we plan on deploying our internal web interface and publishing our promoter catalog on our website for licensing, allowing clients immediate access to our current available offerings. Our goal is to reach at least $500,000 in sales revenue through a combination of co-developed, custom-ordered promoters and licensing from our catalog by Q4 of this year. We're currently raising $2.5 million, where part of the funding will cover the cost to conduct our validation experiments for our potential clients, while the remaining funding will be split between operations, staffing, business development, and legal work. Additionally, from the $2.5 million, we plan to allocate $500,000 specifically towards matching organizations that pair SMEs with large pharmas to co-develop gene therapies. Through these types of programs, we'll need to fund about a third of the project, with the potential that the partner will license our drug later on and take it to the clinical trial. We are a committed team to address the high failure rates and inefficiencies in gene therapy and drug development. Our team brings over 20 years of combined experience in bioinformatics and gene therapy research, with more than 30 publications, 2 current gene therapy patents, and recognition through poster awards at international scientific conferences. We are also guided by esteemed advisors and mentors in Canada, providing both scientific and business expertise. Lastly, we'd like to recognize all the support we receive from various incubators and financial sponsors supporting us throughout this journey. So join us today to help gene therapy developers bring more cures to the market. Thank you.
Thank you very much for the presentation, Luca. We're going to open it up for questions in the chat if anyone has any questions. While we wait, maybe you can kick us off with a question. Is the AI platform able to make promoters that target any type of cells, or are there any limitations?
So the limitations just lie on the raw data that is available. And one thing that we've established as a potential moat for ourselves is to actually generate this primary data ourselves. So my background, I'm doing my PhD right now. I'm a chromatin biologist. So we employ a lot of these types of next generation sequencing strategies where we probe various proteins in mice or in other sample types. So we could build our own library targeting those cell types. So as long as the raw data is there, this model can adjust to pretty much any cell type condition. And part of this catalog that we've published, we've targeted not just neuron-specific targets, but also subsets of different neurons, as well as different brain cell types, heart cells, and so on.
Great. Thanks very much, Luca. And we have one more question. Do you have any example of how well your promoters work compared to what the industry is currently using right now?
So we do have a couple of examples of that, and I've showed a figure comparing one of our neuron-specific promoters to an existing neuron-specific promoter. It's about 3 times as strong compared to that. And one of our biggest selling points as well is our size. So we can offer, our typical range for a promoter is between 180 base pairs to 250. That's kind of our sweet spot area. If you go to commercially available sources like Vector Builder, typically they start at around 300-400 base pairs and up. Our current, I'd say, most exciting offering is a GFAP promoter at a size of about 200 base pairs. We've demonstrated pretty precise specificity to GFAP positive cells in the brain. And an existing promoter that you can purchase, again, from a vendor like Vector Builder, would be the size of about 600-700 base pairs on the low end.
That's great. Thank you so much, Luca, for taking us through your presentation and answering the questions. That is time. If you have any other questions, you can reach out to the team, RePair Genomics, using the QR code on the screen. Thank you again, Luca.
Thank you.